My name, okay, cool, we're recording. All right, my name is Tori, and today I'm just going to give you guys a little bit of a talk about biomanufacturing and synthetic biology. So before I begin, I just want to give you a little overview of my path to NREL. So I'm a um, bit of a younger person here, but I grew up in Washington. And I pretty quickly knew I wanted to get into the sciences with my mom being working in the pharma industry herself. So I went to school at Lewis and Clark and majored in biochemistry and molecular biology. And there I was able to do a lot of wet lab experience and do some of my own research, primarily looking at epigenetic factors controlling stem cell fate. But with that, I gained a lot of tools that enabled me to genetically engineer organisms. And so when I graduated, I was looking at positions and I saw that NREL had some postings for internships, specifically a full year post-bac internship in the synthetic biology group. And so I applied to that and was able to get a position here at NREL, and I worked for a full year doing strain engineering for mucinate production in Pseudomonas putida as part of the Agile BioFoundry. And then once that year internship ended, I was able to then stay on as a technician, which is my current role, just taking on a little bit more projects, but still primarily working on metabolic engineering in the synthetic biology group here. And so if you have any questions, I'm happy to chat more about this later too, just kind of what I suggest or any questions related to internships, happy to chat. So then diving now into, it's a little laggy on my end, but hopefully you guys can see that, the Agile BioFoundry. So this is just a slide to show you what what the Agile BioFoundry is. So we're a consortium of 7 DOE and national labs. Which the mission is to develop biomanufacturing tools and processes to enable sustainable industrial production of renewable fuels and chemicals. And so, yeah, I work here at NREL, which is in Colorado. And then, so just to define some terms, so biomanufacturing utilizes biological systems, usually bacteria or yeast, to convert plant material into commercially important products, such as proteins or chemicals for use in medicines, food, and industrial applications. And so this has been going on for millennia. Humans have made products such as alcohol and lactic acid using bacteria and yeast, making beer and bread and other things. And then synthetic biology is what is more of a modern kind of field that can be used to develop biological systems capable of producing all sorts of other things, including chemicals and fuels, in order to replace hopefully some more fossil fuel derived chemicals and processes. And so I'll be diving more into what that looks like in the future slides. And so just to kind of define some of the type of products that we might do or develop in biomanufacturing. We have what are called bioreplacements or bioidenticals. So these are things that are just trying to replace existing molecules. So things like dairy alternatives, and the idea is to be able to achieve cost parity with those processes. And then there's a lot of new molecules that you can create, which are called bionatives. And so this is where synthetic biology is really disrupting the industry and being able to create, alternative plastics, leather, silk, all sorts of things you can think up of. There's probably a bacteria or some sort of organism that can be engineered to make it. Um, All right. And then so the key kind of ideas or goals in biomanufacturing are again to just create novel products and processes or innovate on those, optimize for resource efficiency. So being able to replace conventional methods, for example, in biomining, that's a huge one that's NREL and other labs are working actively on. And then you can think of expanding the material availability and reducing costs. So there's a huge biomanufacturing can really lower the cost for making a lot of these materials. And then importantly nowadays too, is being able to strengthen our supply chain. So being able to convert different waste streams such as lignin into different compounds and materials. And so these next slides just kind of show the disruptive impact of synthetic biology, which the Boston Consulting Group did some good reporting on, just to kind of highlight where synthetic biology will be in the next 5 to 50 years. And so you can see all these different industries that synthetic biology has the potential to disrupt. And so we're seeing this already in terms of like alternative meats and beauty and being able to produce different products such as squalene oil. You know, fashion, agriculture. There's a lot of different, industries that biomanufacturing and synthetic biology has its hands in. And then looking forward, so now we're kind of at this precipice of where we're starting to see biomanufacturing, synthetic biology really take off. And so there's a lot of products, coming more commercially viable, but I think we're starting to see this kind of these trickle into more the industrial scale and wide scale adoption, and that will only get bigger and bigger as the years go on. And so lastly, this slide just kind of highlights where we're seeing, I think, the strain engineering start to develop. And so we have, again, back in the 1920s, we were, just reliant on the natural ability of these strains and then being able to optimize it, for example, for antibiotic production. And then with the advent of recombinant DNA technology, we've been able to iterate and design new microbial products such as enzymes and biologics, such as insulin. And then now we're really into this digital age of engineering, of being able to produce all sorts of new proteins, enzymes, and molecules just by engineering new to nature sort of proteins with the advent of AI and machine learning. So that's another, I think, important to highlight, just kind of the exciting era we are in terms of synthetic biology. All right, so kind of just to dive more deep into exactly what is metabolic engineering. It's important to kind of distinguish synthetic biology from metabolic engineering. So synthetic biology is really this big umbrella of having, or it provides the tools for metabolic engineering. So you can think of, tools such as CRISPR, computational modeling. These are all synthetic biology tools that we as metabolic engineers leverage in order to optimize metabolic pathways production of the specific compound that we're trying to make. So in our group, and as part of the ABF here at NREL, we're trying to make a compound called mucinate, which is really important for bioalternatives to nylon and tetraphthalates. So hence why we're really interested in making it. And so to understand again how we can control metabolism, it's we want to, or as engineers, we really want to be able to control metabolism. So importantly, if I'm trying to make mucinate, I really want to optimize the carbon flux towards this end compound. And so mucinate being shown here in this pathway, I would want to convert whatever the feedstock that I'm giving it, in this case, sugars, arabinose, glucose, and convert as much of that I can into mucinate, so that the cells are producing it at high yield. And so how do cells do that? So there's kind of two different ways cells control metabolism. They have, on one hand, the gene level. So some of you might know of this tryptophan opteron. It's kind of a great example of how cells control or their gene expression. So if tryptophan is present, you'll start to transcribe the genes related to metabolizing tryptophan. And if there is no tryptophan, then you'll repress this operon. And so that just shows you an example of how that is controlled on a genetic level. And then there's also a protein level, which I won't talk about today, but I just want to highlight because it's exciting with the advent of, again, AI and machine learning. We're being able to now design novel proteins and be able to really, create new processes and tinker a lot more with metabolism on more of this protein level. And so focusing again on just the genetic level, we all need to understand how cells control genes, and this is where the central dogma comes in. So when a cell wants to turn on or make a protein, they first want to transcribe that part of the DNA that encodes for that specific protein or that gene. And so that first process is called transcription. And so the cells, a protein called RNA polymerase will find that gene and then start to transcribe it and create this RNA molecule. And then that RNA molecule then gets translated into protein via the ribosomes. And so as engineers, though, again, we really want to understand how we can control these processes. And so that's where these different elements come in. So you have your promoter in the case of transcription. And so that determines the rate at which RNA is that the DNA is transcribed into RNA. And then in a very similar fashion, we have what's called the R ribosomal binding site, or RBS, and that determines the rate of translation. And so as engineers, we might want to create a library of either promoter sequences or RBS sequences, and that will give us a range of kind of control of either high or low expression levels. And then you would transform that into a cell upstream of the gene you're interested in, measure the expression, and then hopefully you're able to then increase the abundance of whatever the protein of interest is you're trying to interrogate. So, for example, if I'm trying to make a lot of mucinate, I would really want to be increasing the abundance of these enzymes upstream of mucinate, such as CAT-A. And so that just gives you a really high level overview of kind of the work that I do as a genetic engineer. And so in the next slides, I'm just going to show you some other tools that we use in synthetic biology. And so this is just kind of a not a comprehensive list by all means, but just some overview of some of the tools that I've used in my work. And so I'll dive into these individually. So first we have systems biology, which are trying to understand how a cell behaves from a genetic level all the way up to a protein level and then metabolomic level. So genomics is looking at the DNA sequence, and that gives you insight into what genes are present and the potential of this organism in terms of what it is it can create from its metabolism. Transcriptomics tells us exactly what genes are then expressed. Proteomics are then looking at even closer what proteins are there and what are abundant. And then metabolomics tells us what are the metabolites that are then produced in those pools from that. And then lastly, fluxomics looks at the different flux of carbon in those pathways. So these are all tools that we'd be really, that would give us some high-level understanding of how the cell is behaving. Next, we have different evolutionary approaches. So for example, if we get stuck and we want our strain to evolve to be able to grow on a certain sugar or to be more tolerant to a certain product like mucinate, for example, or something that's acidic, we might evolve our strain on that sugar or that stressor. And so we can find non-intuitive targets or just let the cells evolve naturally to be able to mutate and harness kind of the natural evolutionary process. And so you can use directed evolution, adaptive evolution. There's tons of different ways you can set up these experiments and really just let the cells loose and see how they evolve, and then be able to sequence and find what those mutations are. Then you have a ton of different genetic tools, and this is a huge area of research. I haven't obviously listed all of them, but you have CRISPR, which is kind of a big one everyone hope or maybe have heard of. And then there's tons of iterations on CRISPR in terms of CRISPR activation or interference. You have different reporter constructs that allow you to gauge the level of gene expression by coupling the gene of interest to like a reporter, such as luciferase, which is shown here, that make the cells glow. And then again, there's tons of different tools that we leverage in synthetic biology. And then there's obviously a lot of computational and modeling tools coming out, machine learning, that allow you to understand kind of the interaction networks that are going on a protein level or a genetic level. And then being able to use protein machine learning to kind of tweeze out these different networks, because I don't think anyone could be able to fully comprehend the entire workings of a cell and the metabolism by themselves. It's definitely something you need a computer and an algorithm to help you with. And so this is definitely another exciting area for folks if they're interested in more of the computational side of things. And then just to wrap things up, I think it's important to highlight again just the future of biomanufacturing, and the current economic impact that it has. And so there's tons of new jobs that are going to be created as biomanufacturing becomes more mainstream. And by 2030, there are future projections that biotechnology is expected to add 56 billion, and with bio-based products contributed to an additional 291 billion. So I think there's just tons of potential here for folks if they're interested in pursuing a career in biomanufacturing, and lots to see in the upcoming future. And so with that, I think I'll end there. And then if, yeah, folks have any questions, feel free to reach out or follow me on LinkedIn. I'm happy to kind of share my advice on how you can, you know, get an internship or whatever it may be. Feel free to reach out. So thank you.
Tori, does anybody have any questions for Tori? You can come off mute or put it in the chat. Again, you can reach out to Tori for any questions that you might have. But we'll go ahead and move on to our next speaker, who is Yi Chao Han from PNNL.
Yeah. Hello, everyone. My name is Yi Chao from Pacific Northwest National Lab. So happy to see so many students join us today. So here I will give an brief overview of Agile BioFoundry, or ABF. So this little about me. I joined PNL and ABF as a postdoc 2 years ago after completing my PhD at WashU. So my background is in computational biology. I have had the opportunity to apply my expertise in computational biology across several like ABF projects. So I will share a few examples in this talk later. So I copied this slide from Teresa's slide. So what is Agile BioFoundry? So this is a consortium of 7 DOE national labs working together to develop biomanufacturing tools, process, partnerships. So the goal here is to enable like sustainable industrial production of renewable fuels and chemicals. So the 7 national labs include like PNL, Berkeley Lab, Sandia, Unreal, Los Alamos, Argonne, and the Oak Ridge National Lab. So they operate as an like distributed biofoundry in close collaborations, and also collaborate with industry and academia. So this distributed model allows us to leverage the diverse infrastructure and the domain expertise across all these national labs. Sorry, it's frozen. Let me, yeah. So think of the ABF like a subway system. So each subway stop represents a beachhead, a starting point of a biomanufacturing. So ABF's target molecule are like a destination nearby that stop. So while our collaborators go maybe another like other nearby target as well. So rather than reinvent the wheel, our partner can ride their ABF subway to take to a key intermediate and focusing on their resources on their last mile stuff. So in this metabolic map, the gray shows all these potential beachhead, and the green are the beachhead we are currently working on, and the blue are our ABF target molecules. Somehow it's, let me see. Next one, yeah. Over these years, ABF has collaborated with 50 partners on 61 projects. So I can give a few examples I know here. For example, we developed a machine learning method for analysis of mass spectrum product with Angular. And with Anza we developed machine learning method for real-time fermentation optimization. And in the C16 project, we use protein engineering and omics analysis to produce mid-chain fat acid to serve as palm oil alternatives. And in Super Brute Food project, we use omics and biosensor to improve the production stabilities. And I also get involved in a lot of collaboration in academia. I can briefly talk about later. So the ABF has 4 core projects. So focusing on production of 2 sustainable aviation fuel, SAF, and 2 platform chemicals. So you may wonder why we picked this specific 4. So let's dive into each one. So first, ethanol production. ABF developed a novel process called consolidated bioprocessing using anaerobic thermophilic bacteria to directly convert cellulose biomass into ethanol without any pretreatment. So this process significantly reduced the cost. Ethanol is a very ideal byproduct because it can be produced in very high titer with high yield as shown here. And also it's quite easy to separate from water in the downstream process. So more importantly, ethanol is very easy to upgrade. It's a precursor to a lot of compound in SAF or gasoline or diesel. Those like upgrade process has been established a long time ago. Another sub-project is to produce alkyne while the fatty acid using rhodospiridium. So it's the fast-growing oleaginous yeast and become a very attractive host right now for industrial scale production of a lot of compounds. These organisms not only consume like complex C5, C6 sugar mixtures, and those are commonly from like those lignin cellulose biomass. And another benefit is it also tolerates a lot of inhibitors in those biomass hydrolysis. And also it can grow into a very high cell density to over like 100 grams per liters. So because this advantage, ABF use rhodospiridium as one of their hosts since its inception. So over these years of effort engineering, we significantly improved the titer of many many target molecule as shown here. The third one is mucinic acid production using Pseudomonas putida. Putida has been engineered to convert multiple sugar into mucinic acid. This is a very valuable building block that can be used to make large volume chemicals. Those chemicals can be used for direct replacement, or they can be used as like a performance advantage products, such as the polyester, cross-linked polyester, these plasticizers. Excuse me, what? I think something blocked my screen there. Oh yeah. Yeah.
Sorry about
So the last one, the other platform chemical we are working on is CHP. It's also a high value chemicals with broad market potentials. It can be converted to acrylic acid used in super absorbents like diapers. and also a lot of polymers which are used in those carbon fibers as well. So here we use fungi to produce CHP with very inexpensive feedstock. So the current titer is about like 40 gram per liter, and we are still continue to optimize for a higher titer rate and yield. Beyond production, ABF also focuses on the tool development. Here we develop bioprocess, bioengineering, and computational tools to advance strain performance for biomanufacturing of a wide range of products. So we have TA and LCA. We have host onboarding, multi-omics, like transcriptomics, proteomics, metabolomics, and also lipidomics. We have like a metabolic flux control and metabolic flux analysis, whole genome engineering tools like SAGE, CRISPR-A, CRISPR-I, and we have like biosensor, which can be used to build feedback control or use in like high throughput readout, which can be used for screening or other selection process. And we also have automatic lab and a lot of machine learning tools like ART. We can get good recommendation, either like a median component or genetic parts from those algorithms. And here we are also able to perform scale-up work in the lab. So now I will share more about the capacity I focus on, particularly on metabolic modeling, flux analysis, and omics. And as I mentioned earlier in the start slide, rhodospiridium is a key host we work with. There are three genome scale model has been independently developed for this species. And because they are independent developed, they are using completely different name conventions, make it very hard to compare. So I developed a lot of method to convert them into a unique naming space. And we are trying to combine all the knowledge of these three models. As you can see here, the overlap between these three models are not that high. So there are a lot of discrepancy between them. So right now I'm trying to solve this discrepancy and try to develop a better quality consensus model from existing models. So within metabolic model, we can perform a lot of analysis. Metabolic flux analysis, MFA, is one of them. So in MFA, experimentally, we see the cell growing at steady state with those isotopic labeled substrate, here such as the 13C labeled glucose. And we measure the pattern of isotope incorporation in their downstream metabolites using the mass spectrum. So we can calculate how many carbon are labeled from the and get the distribution. So as shown here, like M0 means like no carbon are labeled, like M3 means 3 carbon are labeled, probably for 3 carbon metabolites. And we also have additional measurement like nutrient uptake rate, product excretion rate, specific growth rate. With all those information, we properly balance the metabolic network, and we use computational models to integrate all this data to estimate pathway flux. So with this flux information, probably the golden standard in the field to get accurate flux method, flux value. MFA can help us to identify the pathway bottleneck and figure out the design strategy for enhanced rate and yield. The metabolic control analysis is another tool we developed to answer the question I list here. So how do enzyme and metabolite concentration control the metabolic flux? So in this type of analysis, we use Bayesian inference to estimate parameters in a kinetic model. So from those parameters, we further derived a key parameter called flux control coefficients. So this parameter can help us to understand how the change in enzyme expression at a reference state impact the flux. So the reference state is usually our best performing strain at their corresponding growth conditions. So this algorithm can help us to guide rational strain design. So pinpoint those target either enzyme or like other metabolite, enzyme for O expression or knockout or other metabolite, we can like modify our growth conditions. And finally, we are also exploring like graph deep learning method for metabolic modeling. So metabolic models or metabolic networks can be represented as graphs in multiple ways. We can view it as a hypergraph. For example, nodes as the like metabolites, edge as reactions. So they can be multiple edge between metabolites, and we can have one edge to connect multiple nodes. Or we can view it as a bipartite graph, nodes as both the metabolite and the reactions. Or here, as I show, we can also model it as a mass flow graph. So with node as the reaction and edge as how those mass are flowed. So after abstract them into a graph structure, we can like label nodes, edges, and even entire graph. So we can use those graph machine learning methods to learn the features and extract those meaningful insight to help us do like prediction or validation of string designs. That's all I want to share today. If you have any question, I'm pretty happy to address them.
Thank you, Yi Chao. Does anybody have any questions? Feel free to come off of mute or drop a question in the chat or Q&A. We'll go ahead and move on to our next section with Sarisha. Are you here?
Hello, everyone. I'm hoping all of you can hear me.
Yes.
Okay.
Go ahead.
So my name is Naga Sirisha Parimi. I work here at ORNL. Most people know me by Sirisha. So today I'm here to talk about industry or a national lab and how can and list some points which might help people make a career choice between these two options. All right. I'll start with a brief background about myself. I'm originally from India. I grew up there. I got my bachelor's and master's in biotechnology from a top-ranked university there. And after that, I decided to pursue a PhD, and I came to the US. I got my PhD in biological and agricultural engineering from University of Georgia. Soon after my PhD, I took up a brief postdoc for a few months also at University of Georgia. And after that, I decided to pursue a career in industry. So I got a job at a giant food and agribusiness industry named Cargill. And I worked as a research scientist there for 5 years. So after my career at industry, then I moved on to ORNL as a technical scientist here. So I've been here at ORNL from 2022, so it's my 3rd year running here. So next slide. All right. So I want to start by talking about what considerations one might help one pursue or navigate a career choice between industry versus national lab. The first thing is, of course, your professional interests. Are you interested in core research, business development, policy making roles? What are your professional interests? Also, like, where do you see yourself, say 2 years from now, say 5 years from now, say 15 years from now? Like, what exactly are your goals? That is the first important consideration you might want to make. Then the next thing to consider is, what are your skills and strengths? Like, where do you fit? What would be the best for you professionally, and what skills and strengths you might possess that might help you succeed in one career versus the other. So the next one to consider is, so before moving on to that, I want to say that this is a very dynamic thing. So as you pursue a career, you might develop some more skills and strengths, which might help you either pursue the same path, or you might want to make a career switch. So just keep in mind that skills and strengths are dynamic. The next thing you might want to consider is, what is your personality and lifestyle like? Are you someone who will thrive in a fast-paced research environment, or are you someone who is more interested in solving problems, doing deep dive into problems? And do you like to work on flexible timings? Do you like to have a more structured work schedule? These kind of things are what also matter in making this career choice. And finally, what are your core values? What are your professional values? Excuse me. What are your ethical values? So you might consider all of these before making that career choice. So moving on. Now let's talk about what are some of the similarities that you might find between a career at a national lab versus industry. Just keep in mind that I'm mostly talking from an R&D perspective. So this is more relevant to R&D careers. And also, this is a very generic comparison. So some of these points may or may not be applicable based on the specific organizations you will work for, or the specific roles you might take up at these organizations. So the first thing first similarity that both industry and national lab share is their mostly team-oriented interdisciplinary research focus. So whether it is to develop a product or develop a solution to a problem, you work in teams, you work across multiple organizations, or even within the organization, you work with multidisciplinary teams. The next thing is state-of-the-art research facilities at both of these, whichever organization you choose, is very likely that you will be working on state-of-the-art facilities. The third thing is stable funding. Of course, this is subject to policy changes in case of national labs, or it is, or in the case of industry, it could be subject to market changes as well. So, but keeping that in mind, in this funding is generally more stable than other type of organizations. And then the structured work schedule, for example, like both national labs and industries have policies on work hours, overtime, personal time off, etc. Then opportunities for publishing and filing intellectual property. Again, this is very specific to organization and roles, but in general in national labs, these opportunities are a little more geared towards especially publications, whereas industries, some industries do have it, some industries don't. So keeping that in mind, but in general, you could find such opportunities in both places. And then there's emphasis on safe operational practices. This is again like very high on the priority for both national labs and industries. So that's something you will see, because they want to avoid any accidents or near misses, or they want you to perform every task in a safe manner. Then next coming, both industry and national labs also have opportunities for project management, mentoring, and networking. Again, this is also organization and role dependent in case of industry, but you would have a little more of those mentoring and networking opportunities when you're at national labs. And finally, competitive salary and benefits. So both of these organizations will keep you happy in that way. So these are some of the similarities you might want to consider. And then moving on to differences. So what would help you make this decision of one versus the other? So first thing is whether you're interested in working for a government-funded organization, which is a national lab, or a private or public-funded organization, which are industries. So for example, national labs are funded by mostly government agencies like DOE, DOD, NSF, etc. Then in case of national labs, the research focus is on problems of national interest, energy being one, probably waste to energy or waste to useful products conversion, such things. Whereas the research focus for industry is very product specific, so our service or product specific based on the case maybe. And then the one major difference is between national lab and industry is the opportunities for multi-organizational collaboration, which is more so in national lab versus industry. Again, opportunities for publishing. I already talked about this, that there are a lot more opportunities for networking and publishing and research communication in national lab than in industries. And finally, a slow to fast-paced research, like based on whether you're working on a near-term solution or a more ambitious but very impactful long-term solution. You could be working on slower, fast-paced projects in national labs. But in industry, generally the projects are very fast-paced because you are working in a competitive world where you need to be fast-paced. And finally, like good job security versus no job security in general for industries. So that is something to consider when you're making a career choice between these two organizations. So I want to quickly share something from personal experience. So I worked for this food and agribusiness industry, and what I liked at this place was that I worked on product-oriented research. At that time, I was really motivated to do that kind of research. Then I was lucky to have access to a lot of automation and state-of-the-art facilities. We worked on robotic liquid handlers, which were very cutting edge. And at that time at least, and even now for that matter. And then I got opportunities to scale up some of my research to pilot scale and work on some projects that were already in manufacturing and developing solutions that could improve the efficiency, rate, yield tighter, that kind of thing. Then I got to work on multidisciplinary teams. And finally, I also got opportunities to work to talk to customers and look at the business side of things. What I disliked was, well, I would say I wasn't super happy with, or mostly with fast-paced research with limited opportunities for deep dive and troubleshooting problems. Coming from a academic like PhD background, I would have liked to have a little more time for that. And high confidentiality and limited research communication. So that was one other thing. So I wasn't able to publish. I wasn't able to present my work to the scientific community because of the confidentiality nature of the projects that I worked on. And also limited opportunities for collaboration outside the organization. So given these limitations, I decided to make a career switch, and I chose a national lab career because I had a heavy interest in energy research and solving problems of national interest, opportunities to publish and present, and opportunities to collaborate across multiple organizations. And I also had some personal life choices to make, and career at a national lab helped with that as well. So that was a big bonus there. So I don't know if I have enough time, but I'll try to be really quick about like some things that might help make a better informed career choice. Pursuing internship, talking to professionals working at these organizations, self-evaluation of skills and setting clear goals. All these might help like making that choice between which career path you want to pursue. And of course, like in order to succeed in any career, you need to be dedicated, devoted. Discipline is important. Having a passion and motivation is super important. Having the right attitude and good communication and interpersonal skills. I cannot emphasize how important these things are in order to succeed. Then finally, just some closing remarks. I guess in the interest of time, I'm going to like skip the slide, but I've covered most of these points. But in summary, it's like what kind of impact you want to have and what kind of research environment you want to work on, which should help you choose this career path between industry and national lab. Thank you for your attention. I'll be happy to take any questions.
There's a question in the chat. It says, is it common for postdocs in the national labs to get more permanent positions after the 3-year contract?
I think it is very group specific. In some cases it does happen, in others no. So it really is very specific to the timing, whoever has hired you, whether there's an open position at that time. So yeah, I guess the short answer is it depends.
Right. No, I would agree. And then there's a longer question here from Teresa Sersha. So maybe if you have a look at that while we hear from Joanna. So thank you. That was great. Joanna, are you ready?
Yes, I'm ready. Hi. Hi everyone. So in my talk today, I'll be doing a brief comparison for those of you who are navigating at the moment career choices with the focus on a comparison between academia and national labs. So whether you're just starting to think about your next step or already leaning one way or the other. So I'm hoping that the few slides I have prepared will give you a clearer picture of what each path might offer. You will see some overlap with some information that Sirisha has already provided in her slides, but here in comparison with academia. So I would like to start by briefly introducing myself. So I'm Joanna Tannous, an early career staff scientist at Oak Ridge National Lab. I'm a fungal synthetic biologist, and my work within ABF focuses on developing advanced genetic tools for industrially relevant fungi to be able to expand their capabilities to produce biofuels and biochemicals. I did a PhD in pathology, toxicology, and genetics at the University of Toulouse in France before I moved to the US. And over the course of my career, I've completed 2 postdoc positions in the US. One, I did it in academia at the University of Wisconsin-Madison. And the second I did at ORNL when I joined the lab in 2020. And that was right before I transitioned to my current research staff role. So based on my personal experience working in both academic and national lab settings, I've broken my talk today down to focus on a few key areas that I personally think are important when you're considering or comparing these 2 career paths to make a decision on which one to choose or which one to go for. So I'll be mainly touching on the research focus and environment. I will talk about career development, opportunities, and job security in both settings. I will compare the work-life balance and culture, funding and compensation. And at the end, I will wrap up with a small take-home message to help you reflect on what might be the best fit for you. So starting off with research focus and environment, which is often one of the biggest difference between academia and national lab. So in academia, research tends to be more fundamental. There's a lot of autonomy in choosing the topic that you want to work on. And usually it is based on what the personal investigator's interests are, or if any like curiosity-driven questions the PI might have. Whereas in national lab, there are more mission-driven. So the research is usually aligned with national priorities, what Shirisha already mentioned, like energy, defense, and environmental sustainability. In academia, also projects are typically smaller in scale, and they're led by individual researchers or like small lab groups. Whereas in national lab, we see more, the research is more team oriented and involves like large interdisciplinary collaborations within the lab. So the access to labs, larger scale facilities are also a little bit limited in academia. And also here I want to move to the second point, and success is often depends on publishing and securing competitive grants. Whereas in national labs, we have access to state-of-the-art facilities and infrastructure that you wouldn't typically see it, or they're not like so common to find in a university setting. And while publications are important when we're working national labs, there's also a strong emphasis on patents, reports, technology development outcomes compared to academia. So now I will move to talking about career development, opportunities, and job security, which I believe it's a topic all of us are interested in when picking a career path. So I'll start with like career progression. So in academia, usually it goes from postdoc, assistant professor, to tenure if everything aligns. Where in national labs, it's more like postdoc, staff scientist, senior staff scientist, or leadership roles. So when it comes to postdoc position, I think someone already asked this question. So they're both temporary positions, either in academia or national labs. But in academia, moving forward really depends on landing a competitive faculty role. In national labs, there's often a clearer pathway to transition into permanent position within the same lab, which can make things feel a little bit more stable. But keep in mind, it's not always the case, and spots can be limited. They usually tend to not like duplicate skill sets, but if you have a unique skill set that can benefit projects or initiatives, I think there's a clearer path to transition to permanent position. Now for job security, academia is very grant dependent, as I mentioned earlier, and tenure track positions can come with a lot of pressure. Whereas in national labs, it tends to be more stable overall, thanks to federal funding, though they're still also sensitive to policy and budget shifts. So in academia, we see that you would have the chance to mentor students and build a teaching portfolio, which is great if you really enjoy that aspect, besides doing research. In national labs, we also have mentoring opportunities, often through like summer interns. There's a lot of programs that we recruit summer interns through, like graduate students, postdocs as well. But there's much less emphasis on teaching. Which is like the big difference. And finally, in terms of professional development, in academia is often self-driven, so you need to seek your own opportunities. Whereas at national labs, there are typically more structured options like specialized training, collaborative programs that are built into the work environment. So now I'll move to talk about comparison when it comes to work-life balance, which can really shape your day-to-day experience in both settings. So first, if we look at the work schedule, in academia, hours can be long and unpredictable, especially when you're juggling both research and teaching and administrative responsibilities. Whereas in national lab, usually we have a more structured schedule, often sticking to standard business hours, 9 to 5. So evenings and weekends are more protected. Academics often struggle to take real breaks because of the constant pressure of grant deadlines, publishing, and teaching's preparation. At national labs, there are typically like generous government benefits that include paid time off, holidays, different types of leaves, which makes it easier to actually step away and recharge. And lastly, for the culture, in academia, the culture can vary a lot depending on the university, the institution, the department, or even just the PI itself. So you will see differences within the same institution between one lab and the other, depends on the PI. So some places are great about balance, others are not as much. National labs tends to emphasize work-life balance more consistently. So there's like formal policies on things like overtime, flexible schedules, and leave, which helps set clear expectations across the board. And now I'll move to talking. I think that's my last point of the comparison about funding and compensation. So starting with funding sources, in academia, the research is largely supported through competitive grants from agencies like NSF, NIH, and private foundations. But yes, they're also eligible for grants from government funding agencies like the Department of Energy or others. In national labs, it's primarily government funded through DOE, DOD, NASA. Yes, they can also apply for grants from agencies like NIH, but it will be like mainly depending on the scope of the work you're applying for. So the funding landscape, I can say that there is an overlap between both, but the mechanisms often vary. As for how the research is funded, in academia, funding is usually tied to the PI. That means researchers often need to secure their own grants to fund their lab, staff, sometimes even portion of their salary. Whereas at national labs, while grant lending and proposal works are critical, many positions are supported through more stable, mission-driven funding, which reduces the constant pressure to secure personal grants just to stay afloat. And when we talk about compensation, academic salaries can vary a lot, also depending on the institution and how well funded the PI is. So they tend to be more modest overall compared to national labs, where salaries are generally more competitive, often higher than academia if we're comparing similar career stages. And they come with strong government benefits, which adds to the overall financial stability. Sorry. So just to summarize and wrap up, if you're driven by passion for teaching, academic freedom, and exploring fundamental questions, I think academia may be your calling, but just be prepared for intense competition and challenges for securing funding. On the other hand, national labs offer stability, top-tier facilities, and impactful and mission-driven work, though the research may not always align with your priorities, but mainly with national priorities more than like your personal interest, I mean. But ultimately, let's put it this way, I think the best path depends on where your passions and values lie. So similar to what Sirisha concluded when comparing industry and national labs. And with that, I would like to thank you. And I would like to take any questions you might have, or if you have other questions or something I did not cover in my slides, if you have other questions, please reach out and I'll be happy to answer.
Thank you, Joanna. There's actually a couple of questions in the chat. If you could have a look at those while we move on to the.
Yes,
Okay, great.
sounds good. Yep.
Again, go ahead and drop any questions you have in the chat or in the Q&A. And we're going to move on to our postdoc early career panel. Tatiana, are you here? There you are. Tatiana is at Argonne National Lab, and she's going to be our moderator for this session. So Tatiana, it's all you.
Thank you, Andy. I'm just gonna check. Alicia, is there?
Yep.
Austin? Okay, good. Sanmi and Apostolos, sorry.
And Austin, yeah, everybody's here.
Okay, great. So I think before we start, I just want each of the panelists to introduce themselves. Maybe if you can turn the camera on so people can see you, it would be great. But if not, it's okay. And then just mention about what are you, your profession or title at the different national labs, and also maybe to share something fun, a fun fact, like a hobby or activity show, whatever you want, just to break the ice. Go ahead. I'm gonna start here with Alicia. Please go ahead.
All right, thanks. Can you hear me?
Yes.
Great. So yeah, my name's Alyssa Bleem. I'm a research scientist at the National Renewable Energy Lab. I got my start here actually by doing a postdoc, and then I had a chance to apply for a full-time staff position. So now I'm a staff member here at NREL, and I do research on synthetic biology, tool development, process development, basically to engineer microbes that can make useful chemicals and materials from waste and renewable feedstocks. And a fun fact is I've been taking fiddle lessons recently. It's been really challenging but fun.
Good. Thank you. Okay, let's go with Austin.
Hey, everybody. My name's Austin Carroll, and I work at Oak Ridge National Lab as a technical professional. So I did a postdoc there in 2019, and then after about 2 years, joined permanently as technical staff. So I work at the bench on a bunch of different projects in the ABF, as well as some other groups in the national labs, including CBI and Bottle. And likewise, I work on synthetic biology and upcycling waste and producing new plastics using microbes as a platform. And then a large part of my emphasis is on domestication of non-model microbes. So taking things that nobody's ever worked with that have phenotypes that are interesting and figuring out how to get DNA into them and engineer them like we do all our favorite model microbes. I don't know if this is fun necessarily, but I'm expecting a new baby girl here in the next 2 weeks to 2 minutes. So if I disappear, that might be why. Thanks.
Yeah. It's hard, but it's fun. Okay, I'm gonna go with Sanmin. Go ahead. Sanmin. Maybe we go with Apostolos first then.
Yeah. Hi, I'm Apostolos. I'm a postdoc in Lawrence Berkeley National Lab, and I work generally on the intersection of machine learning and synthetic biology. And I focus a little bit more personally on process, and I try to move towards scale up. And I'm a chemical engineer by training, and I switched to the AI focused position for my postdoc. And a fun fact is that I've been getting back again into martial arts, which I very much enjoy post PhD when I do have the time and energy to focus on anything else other than work.
Great, thank you for sharing. Okay, Sanmin, are you ready? I see you're connected, but you're muted.
Can you hear me?
Yes, yay.
I'm sorry. Yeah, hi everyone. My name is Sang Shin. I got a PhD majoring in microbial physiology and biophysics from the College of Biological Sciences at Seoul National University in South Korea. I'm currently working on protein engineering as a staff scientist at Los Alamos National Laboratory, New Mexico. One fun fact about me is that recently I participated in a project called Bottle Consortium, which focuses on plastic degradation, especially using microbes. I believe that one day humanity will solve this plastic degradation problem using microorganisms. As you know, microbes are everywhere, but I found myself worrying, what if one of those engineered microbes accidentally escapes from the lab and degrades all the plastic products? It will cause global chaos. This concern is still very present for me. Outside the lab, I've been a big fan of the Queen since I was a teenager, especially Bohemian Rhapsody. A few years ago, I suddenly wanted to play this song and started learning drums. I'm still a beginner, but it became one of my favorite ways to relieve stress. So if any of you are looking for a fun stress outlet, I definitely recommend trying playing drums. Nice to see you all again.
Thank you. So I'm gonna ask a question, but the idea will be that all of you participate. So just to break the ice, I'm going to ask, like, I think some of you already answered these, but how you end up here. But my question is going to be more like, what are the skills or talents that you have discovered through these years working in your positions? And what are the things that you think that I wish I knew this before?
Sure, I can start again. So I think maybe one thing that really helped me coming into my position now is that I had a lot of really diverse experiences. So my PhD was doing a lot of computational work and it was in the context of healthcare, but my undergraduate was in chemical engineering. So I knew a lot about process engineering. So basically, when I came to NREL, I was able to speak the language of a lot of different fields. And that was really helpful for me. And I think it still is. One thing that I wish I had known a little bit more about was techno-economic analysis and process assessments. I think that it is very important, at least when you're in a really applied space like the national labs. You kind of have to think about how your technology fits into the whole process. And it took me a while to kind of start thinking that way and learn how to kind of integrate with those people early on.
Who wants to go next? Okay.
I can go next. So a little bit on the more technical part, there is this field of Bayesian inference that I was not aware of. And if you're working with any computational part or parameter inference, that is very, at least something to take a look at, because it allows you to know what you don't know. And sort of this kind of extends not only on the computational part, but also uh, on the general part of your work, where it is a very good skill that I'm still learning how to do on figuring out what are the things that I'm missing and to try and find people to ask the right questions. And the National Lab sort of allows that a lot because there are very skilled people next door, which in academia, because it's a bit more lonely, is not as encouraged. And you kind of, so the one skill is go and ask questions of people that are around you if you have access to them, and the more access you have to them, the easier it becomes.
Yeah, I completely agree. When I was doing my PhD, I felt very lonely, and I went to national labs, and there is so many people with so many skills or knowledge that it was completely different. Okay, Austin, do you want to go next?
Yeah, certainly. I kind of going off of that, coming from my PhD to a postdoc, I think I had learned a lot more than I had realized as a PhD student. And so I got there and, you'd be given tasks and I'd be like, okay, well, who's the expert on this? I should go talk to them. And I think one of the big things you realize, especially as a postdoc, is it's you. You're the person kind of at the edge of the knowledge base. And I think when you get into that position, the sooner you realize that, kind of the better. The sooner you stop looking around and you start kind of looking inward and can act with a little more confidence in terms of like guiding the research and designing experiments. And I think that kind of flows downstream into like how you navigate your career. It kind of comes back to you. You have to decide what you want to do and kind of what steps you're going to take to get there.
Sanmin, you want to answer this question? Maybe you're muted. Okay. I see a question in the chat, and this is for NREL. Maybe Alicia, you already answered, but is there a good emphasis on chemical engineer at NREL?
Yeah, I did answer that in the chat. Definitely, so at all the national labs, I think there's a lot of engineers and a lot of chemical engineers. At NREL in particular, we do a lot of process design, sustainable process design to make chemicals and fuels and materials. So that's very applicable for chemical engineers.
Thank you. The next, somebody have any question? Feel free to unmute yourself and share.
I'd like to follow up on the chemical engineering question because I was in a small conference about synthetic biology, and the thing that I realized is that there were more chemical engineers than biologists in a synthetic biology conference. So even also on the process side, but also on the biology side, there is a lot of chemical engineering going around. So there is definitely a focus on that.
Sanmin, I see you unmute. Do you want to share your skills and what are the things that you have learned through your job?
Oh,
Yes.
hello?
Yes.
Sorry, I'm really sorry. What was your question?
Is there like, can you share what are your skills and what you have learned through your career in the national lab?
Oh, yeah. So, yeah, basically, in my career engineering, experiment don't go always work out. So I had to get better at setting realistic goals and planning closely with my colleagues. I believe National Lab, the benefit of National Lab is collaboration with others and even other field. So yeah, I learned a lot of the collaboration between people here.
Thank you. So I have more questions, but I want to give you the opportunity to ask directly. So please feel free to unmute. or drop it in the chat and I can read it. While somebody finds questions, I will ask another question. How your job evolves, if it evolves, and how do you, okay, I'm gonna pause my question and I'm gonna let JC Taylor to ask. Go ahead. And if I say your name wrong, please apologize.
Yeah, thank you, Tatiana. This is Jace Taylor here, and I wanted to follow up with Austin. Hi, Austin, also former Atsumi member here. And yeah, I just wanted to follow up on your comment about becoming the expert as a PhD and sort of looking inward on, your knowledge rather than trying to look elsewhere to answer questions. And I was just kind of wondering, how was that transition from working with, model organisms to working with non-model organisms, and if there was a learning curve that you had to consult other people for, or if that was something you had to figure out yourself?
Certainly, yeah. So it kind of depends. The definition of model can be pretty flexible. Some people would consider like cyanobacteria to be non-model, or at least that was kind of the thinking when I was in the Sumi lab. Now maybe they're more model, maybe they're not. It depends on who you ask. So it's definitely a gradient. Eventually, where I ended up was we're pulling things out of the soil in my backyard and building tools for those, and then anything goes there. There are definitely like touchstones along the way. Everything has some genetic relationship to other things. So you can kind of look at guide stones, and even people within our lab have expertise in similar enough organisms. So you kind of use that as a jumping off point. Um, but really, especially in the Gus lab, we kind of have developed a system of or a way of doing this systematically. So taking a similar approach for all of these different organisms. And so there's been a lot of collaboration with other people in the lab on, okay, we're not working with the same system, but what's the general approach you took? What were the tricky parts? What parts were easy? How are our microbes similar or different? And sometimes that's very fruitful, and they'll give you physical things like DNA plasmids to go and try. Sometimes it's a little more esoteric. It's a little more of like general approach stuff that you can kind of pick and choose what actually applies to what you're working with. So yeah, that's to say you rely on others' expertise. Like I think somebody else had mentioned that as well, especially at the national lab. You do have this huge breadth of expertise among everybody else. But then you have to take that and look at your project or what you're working on, and make the calls as to where their advice applies to what you're doing, and where maybe you need to deviate from that, or go look for other sources, or just start testing things kind of on your own.
Great, thank you so much, and congratulations on your daughter.
Yeah, thank you very much.
Great, thank you. I see there is a question in the chat about what kind of research in microbiology we do at Argonne. I'm not the expert in that area because I'm more on the analysis at Argonne, but we have a lot of research in the microbiology, and there is a division, the bioscience division, that focuses on that and has a lot of technology and portfolio in this area, in the biomedicine, and also in genetic modifications and things like that. I can send you a link where you can find more information, and if you have a specific questions or if you want to connect with somebody, I'm happy to help with that. Is there any other question that people want to ask? I want to then ask about what's the biggest challenge that you have faced in your job, and how you like resolve those challenges, and what you learn from that.
I could start with this.
Go ahead.
Other than technical parts that are hard, and generally you get to figure these things out. Organizationally, one of the hardest things to do is sort of focus on work because there are so many interesting things happening here and there. And you hear about a cool project or a cool meeting that's happening or something that you want to be involved in. And because it's so collaborative, it's easy to sit down and talk with someone for 3 hours and come up with a project that should take 2 weeks to try out, sort of situation. And then because there is some flexibility as long as you go along with a mission that your lab or your institute or all the national labs have, you can try things out and you can experiment. So this, you have to tell yourself and others no more often than you want to. At least that's in my experience, because I get distracted very easily too. Uh, so yeah, I've been here for 2 years and I'm still learning how to not get distracted by shiny things around me.
Maybe
Yeah,
if I'm
I would actually totally echo that sentiment and say like getting spread too thin is probably the biggest challenge I've faced in learning to focus on things. I think at least for myself, like the best part of a project is the first 3 hours where it's new and you have all these ideas and they're very exciting. And a lot of them are just very exciting because they haven't not worked yet. And once you hit that part, that's kind of where the real work begins and where it gets challenging. So being able to anticipate like what your actual your capacity is, not in terms of like skill or anything, but just physically what can you accomplish in a given time frame, and realistically what are the most important things and where you think you can make the biggest impact for a project, and then likewise balancing that with like what projects are challenging to you, and are you going to learn something very new from, finding that balance. I'd say is probably the hardest part, at least for myself, in terms of navigating a career in research.
I think I would just build off of both of these comments, which are, yeah, I agree. For maybe for folks on the call who are considering doing a postdoc after their PhD, one transition that I found that was kind of challenging was in your PhD, right? You're good at pretty much one thing. You have like one project, it's your thesis, you're like fully in it. If you come to a national lab, at least for a postdoc, you're probably going to be on more than one project, and there's probably going to be multiple elements to each project. So you have to figure out, okay, I can't just focus on this one project anymore. I have to arrange my time to advance everything, but kind of like spend enough time focusing on each thing that I actually get things done. So for me, that was a really big change. And I think it took probably at least like 6 months to get organized and really feel efficient working on multiple projects.
Yeah, that is completely true, that you start becoming more efficient and prioritize, and you learn like a method, and then you know how to tackle the project next time. I like to share that a challenge, but it's not about, it's a very good challenge, is that in national labs we have a lot of nationalities, people from around the world. So you have to learn how other people react to certain things and get like understand the cultures, the diversity, and that is very interesting, and it's a really nice learning process because you start becoming a more patient person to understand other people emotions or feelings or thoughts, and then you adapt to that, and you realize that it's not just one type of thinking but many thinkings that at the end will help solve problems in a nice way, very creative way. So I found that very interesting. I remember coming the first time to the US that like in Latin America, we're very close, we're like very like loud, and then talking to certain people from other regions, they were like, hey, you're like a little too crazy. Then I learned that, okay, I have to manage a little bit more of my emotions, but that was very interesting. Okay, Min, are you there? You want to share your thoughts about this question, about the challenges? Okay.
Sure.
Yeah, go ahead.
Good. Thanks. Yeah. Um, since I'm still early in my role as a staff scientist, so two big challenges have been project management and the scientific writing. I'm really want to talk about writing skills. So, writing small proposals to show my ideas clearly to the reviewers takes a lot of practice. Uh, it's something I'm still working on, and then it doesn't come just overnight. But yeah, it's a crucial skill in this field.
Okay. I see there is a question. Dhruvi, do you wanna ask your question?
Hi, yes.
Go ahead.
So really nice. This is a really nice meeting out here because it's giving a lot of, insights about National Lab, especially for a person partly from academia and have worked a little bit in pharmaceutical industry back in India. So my background is, like I said, pharmaceutical background with drug delivery, but currently I'm in biomedical engineering and I'm working on developing contrast agents for MRI for breast cancer, due to which, like, currently I have, imaging background as well as, all the in vivo models for cancer and genetic modifications of cells and everything. So I would just like to understand, coming from this kind of background, what labs to look out for and what kind of research is going on currently that can, fit with my background.
Yeah, I can speak to NREL at least. So
Uh-huh.
obviously, like NREL is the Renewable Energy Lab, so we don't do anything with animals or health or things like that. But there is some imaging here. It's not a huge amount, and I don't know a ton about it. But in the Bioenergy Center, the center that I'm in, we have a small team of folks who do imaging for biology. So there's like
Sure.
electron microscopy, and we have some cryo-EM expertise, and things like that. It's a pretty small team, but I do have certain projects related to biological processes and then also like deconstruction of biomass. So they can look at different feedstocks and like how they break down and things like that. The other
Gotcha.
kind of area where we have a lot of imaging is in kind of the more solar and materials space. So those people aren't looking at anything that's alive, but they're using advanced imaging techniques to characterize materials for energy applications. So I'd encourage you to look at probably that sector of NREL if you're interested in imaging.
Gotcha. Thank you so much, Elisa.
Sure.
And thank you, Angie, for the resources, and Tatiana too.
I think that generally, if as a rule of thumb, if you're doing it, somebody in a national lab also doing it. From what I've seen, there is a huge breadth of things that's happening, and so there are people doing either that or data analysis for MRI and all of these imaging techniques. But I am unfortunately not very well equipped to give you anything other than what I just saw from Angie. But I'm pretty sure you would be able to find people doing things adjacent to what you're doing in at least one of the national labs.
Gotcha. Yeah, I'm also like at the side, since AI and machine learning is on boom, but I am a pure wet lab student. I'm also trying to get like a certificate course in like, analysis or imaging analysis using AI and machine learning, because it's just, it's just a boom on a boom right now. So maybe by the end of this year, by the time I graduate, I might have like one of those skills too. So something like that. Okay, sounds good. Thank you so much, Apostolos.
Great. Any more questions? I have ton of questions, but I want people to have this opportunity to interact with our researchers. Maybe I can ask the last question before we end the panel. It's what kind of advice will you share for people that are interested to go to a research career? Could be a national lab, or based what we learned from like today, could be any other area like industry, or based on your experience basically. But
I can start this off too. So I don't remember which one of the speakers mentioned this, but interpersonal skills is the like supposedly technically you're gonna tick all the boxes and you're gonna be the expert in the actual thing that you're doing. But especially within the national labs where there is a collaborative environment and you're not alone in a lab just crunching away at a bench, uh, being able to sort of read other people and uh, convince them and communicate in an effective way, but also being emotionally intelligent about like what the way that you're talking, how it affects other people, and this could make or break collaborations and make or break the environment you're working in. So the number one thing is kind of keep an open mind and keep your antennas out so that you can actually interact with other people in an emotional way too, and make everything in your work life easier if you're interacting with other people.
I completely agree.
And also, sorry, to follow that up with, I was taking a writing workshop recently, and like this also shows on the writing side. So what Sangmin was saying before, that it's you need to communicate your ideas in writing as well, and that's hard. That also applies on writing, so it kind of extends in every aspect of communication.
Yeah, I can go off of that too. Just to echo the writing sentiment. I probably every 3 years will go back and read something I've written and be like, this is the worst thing I've ever read. Like, why would I even phrase it like that? What was I trying to say? So you're always getting better. But I think a lot of that starts with being critical of like your own writing. But that in a fun way can start by being critical of other people's writing. So reading a lot of papers and deciding which ones were very well written and which ones you have to read the sentence 5, 6, 7 times to even understand what they might have been trying to say. Um, I think that's kind of where getting better at writing starts is reading a lot of papers and thinking about them kind of from the other end of like, okay, how would I have phrased this? Or, what's a more concise way? I think that's a huge thing that at least for some people I've talked to, as well as myself, kind of goes under the radar in grad school a little bit. Nobody teaches you exactly how to write. It's kind of just assumed that you've either figured it out or, best of luck to you. So that's one thing. Another piece of advice is like, there's a lot more to a career than just the job. This will be my third kid, and coming to a national lab and choosing the position and the PI I chose have a lot to do with that. Somebody who's flexible and who would, I felt like I could do that and still accomplish things and get things done. But then also like in terms of choosing what kind of position you want at the end of the day, um, research is a lot and is very important to me, but it's balanced with a family on the other end and stability. And so, at least personally, that was what was so attractive about the national labs rather than going into industry where, I could come here, start a family, buy a house as a postdoc. Whereas in industry, especially kind of where we were at in California, there wasn't really much of an opportunity to do that. It would be an apartment with maybe one kid rather than a house with three. So, it's hard to get advice on that in the lab and from your mentors, but if you find the right one or you ask around, you can find that. And that stuff probably is 50-50 with what the research I'm doing is in terms of like how my career path has gone. So just I guess maybe something for people to think about, and to let those things be important to you if you feel that they are.
Yeah, kind of building on that, like try to enjoy what you're doing. I think a lot of us in science think that, okay, I'm gonna get my degree, then I'm gonna do my PhD, then I'm gonna do a postdoc, and then I'll be a PI. And there's like this linear trajectory. And I've seen so many people, colleagues of mine, change, and it's because they're not enjoying it anymore, right? So you want to enjoy the research topic or the types of people you get to work with, the kind of applications. I think we're really lucky in the national lab system to be pretty like mission driven. So a lot of the people that we work with really care about what we're working on. But sometimes you might not enjoy like the day-to-day workflow. It just doesn't jive with your lifestyle, right? So we've had folks in our group change from being in the lab every day to being analysts. And now they're doing modeling and they come in once a week, and it just works a lot better for their schedule. So you can always change your situation. And I think it's important to remember that and take advantage of the resources afforded to you by doing a career in science. There's a lot you can do with it.
Okay, Sunny, you wanna finalize the panel?
Yeah, I'd like to talk about the wet lab work. I think basic lab skills, even for the computational biology work, necessary, like growing microbes, especially in microbial field. Growing microbes, extracting DNA or proteins, keeping things sterile during the experiments is also helpful to know how to organize it and analyze your data using tools like Excel or something. And as mentioned earlier, we need to keep being a good team player, staying curious, and being willing to solve the problems when things don't go as expected. A lot of work with questions and trying to figure out the answer. That's my opinion.
What was the last thing you say? Sorry.
Excuse me?
What was the last thing you said?
One last thing. Sorry about that. What was the question?
The last thing you said?
Oh, yeah, so yeah, our work starts with questions, yeah, and trying to figure out to figure out the answer. It's the basic of the science.
Okay, great. Thank you so much for our panelists. I think that we are on time for the next activity, Angie, right?
Yep.
Yes, thank you so much for your time.
Thank you to Alyssa, Apostolos, Austin, Sangmin, and Tatiana. Appreciate all of you. If you could just stick around for a couple minutes in case we have any questions come up in the chat, I'd appreciate it. Okay, so our next topic is professional development, and that will be presented by myself and Karuna Jetty. Real quick, my name is Angela Tarver. I didn't introduce myself before. I'm the program manager for Agile BioFoundry. So I help on the back end with coordinating, reporting, organizing meetings, all hand meetings for our 140 plus ABF staff, and kind of everything that goes with that. So I'm going to start on our professional development section, and then I'll hand it over to Karuna. So we're going to go over a little bit about where to find job postings and available internships, some do's and don'ts for resume writing, how to put your best foot forward when you're interviewing, and also how to best network and kind of get your feelers out there to really see what's available for you. So first is job postings. From our Agile BioFoundry website, you can find job postings by clicking on Work With Us and then Careers. You can also find specific job postings at the different national labs. Because we're recording this, we'll make all this available as well as the slide deck so that you will have all these links. So don't try to furiously write them down. We'll definitely make those available for you. Internships, I'm going to go into this one a little bit more in depth. You can also find these on the Agile BioFoundry website under Work With Us and Internships. So on that page, you'll see a couple of testimonials from previous interns. I believe Tori is actually on there as well. She used to be one of our interns. And then on the next slide, you'll see that, we have the links in for different internship pages at the individual site here, and we'll also make all these links available for you. But I'm going to go a little bit more in depth with these few internships that are currently available through DOE at our lab. So the first one is the Science Undergraduate Laboratory Internship, or SULI. It's the most common internship across the ABF National Labs. And just a little bit about that, you'll be mentored and guided through a specific research area to gain exposure to many of the science and technology areas that are the foundation of the Department of Energy, or DOE, mission. SULI is a paid internship opportunity sponsored by the DOE Office of Science, Office of Workforce Development for Teachers and Scientists, or WDTS, in collaboration with DOE laboratories and facilities. Students can participate in SULI either in a 10-week or 16-week internship. The second one is the Community College Internship, or CCI. This program is similar to SULI, except for it's available for community college students, and typically only offers a 10-week internship. And then the last one is the Science Graduate Student Research Internship. This is specifically for graduate students. This program creates a pathway for you to advance your PhD thesis research while working at a DOE national laboratory, collaborating with scientists using state-of-the-art facilities and cutting-edge scientific instrumentation. So let me go to the next slide. So all of these internships are currently open and taking applications. I'll make sure that everybody gets these links. If you had registered for this workshop early on, I had sent out some information about different application workshops or informational seminars based on these different internships. So if you didn't receive those, if you go to these different links, you'll be able to see all the information available for these internships. So next is resume writing. So resumes or CVs are different. So resumes are typically job specific and usually only 1 to 2 pages. CVs are typically all of your technical experience. So first, be careful what the position is asking you for. But if you're just doing a resume, those you can tailor for whichever job you are applying for. You can use keywords or phrases from the job description to align your experience. You want on a resume only provide relevant information. So don't just tailor your resume, but definitely keep it relevant to the position you're applying for. Incorporate keywords naturally, so include job-related terms, but don't overstuff your resume with irrelevant keywords. If the keywords are incorrectly used, your resume will come across as unfocused and insincere. Use a clean professional design. Unless you're applying for a creative industry that expects more, choose a simple format with professional fonts and consistent spacing. One of my biggest things is proofread. Make sure that things are spelled correctly, using the correct grammar, because this is your first impression for a position you're applying for. You definitely want to put your best foot forward. And then you definitely, something that Apostolos had spoken about is emotional intelligence. So this is what we call soft skills. So you definitely want to balance hard skills, so more technical lab bench or analysis type skills with soft skills. So that is also important. Things that you shouldn't do. Don't include irrelevant work experience. If your first job was at a Dairy Queen, probably doesn't apply to a national lab position. So probably don't include something like that. Don't include unprofessional email addresses. Something simple like first name, last name at gmail.com or something along those lines. Don't include personal information. So don't include marital status, nationality, or other details that are not relevant to the job are important. And I think the biggest don't is don't lie or exaggerate. It's not worth lying about your education or your experience or anything else. Typically through references, hiring managers will verify your claims, and hopefully they won't find any false information that you've provided. And I think I'm gonna, at this point, hand it over to Karuna.
Thanks, Anchi. Hi, everyone. My name is Karuna. I'm currently a postdoc at Sandia National Lab, and I have been working with ABF for the past 2.5 years. And my background includes metabolic engineering and with a focus of bioproducts and biofuels. I think we have around 8 minutes, so I'll just talk a little bit about interviewing and networking. So what to do and what not to do in the interviewing process. The first thing is to research about the company or the lab which you are applying. And the research should include looking at about your the company website or the interviewer LinkedIn page or also the social media accounts, because this generally gives you the background information. And it's also a good idea to search about the recent industrial news. This is because to bring up a talking point or something. These kind of efforts show that you are really interested or like you are passionate about the job. And the next one is to prepare answers to common questions. So it is a great idea to come up with a list of examples of times like, you have success or you showed leadership in the past. So when you're answering this kind of questions, you generally have a STAR method.
means like star denotes like situation, task, action, and result. So know each of these 4 components for your examples, and you can adapt them to each question. And if you have any gaps, like work gaps in your work history, you have to be prepared to cover them up, and you have to have a genuine reason to answer for that. And the next one is to ask questions. Interviews are generally like a two-way street. So it's always fair for you to ask questions to the interviewer. Oftentimes, these interviewers will end up the interviews like, asking, do you have any questions for us or something? So it's always better you have a set of questions. For example, like, you can ask them, what does a typical day look like? Or what are the projects I'll be involved? Or you can even say like, what training programs are available for the employees and anything. And a quick search of these all stuff in Google, you'll get a multiple questions, I guess. And the last one is the follow-up. We all know for every job posting, we have a lot of candidates, right? So to stand out from the crowd of interviewers, so send up a follow-up email to your interviewer, just thanking him for his time and an effort. And that really makes a difference, I guess. And what are the don'ts? The first and foremost, never be late, because that doesn't go well with your attendance record. We always recommend, getting to the interview at least 15 minutes before the scheduled time. And as the saying goes, if you're on time, you're late. We never know the unexpected conditions like, traffic jam or something else, and you may be late. So it's always better to be on time, like, 5 minutes or 10 minutes. And next thing is, do not dress inappropriately. That means whatever you make, whatever you wear really makes an impression on the interviewer. Don't underdress, because that will show a lack of professionalism, or if you overdress, it may be distracting, or too many accessories or anything. So what you wear to the job interview really depends on the industry and the position you are looking for. And the next one is, and the most important thing is, never speak negatively about your current or your past employer. Because, when you speak about them like that, it doesn't win any brownie points for you, but it gives an impression that you may be a risky hire who may spread some negative information or blame others from some problems which are occurring in your group or anything. So it's always better to give some diplomatic responses about your past employees. Like, even if you had a bad experience, or if your tenure just ended badly, it's always better to give some diplomatic answers and try to find something positive to say about your past work experience, and then move the conversation along to highlight your skills, so that that would be beneficial to your future employer. And the next one is, never get distracted. The full focus should be on your interviewer. So phone distractions, like it shouldn't be on the ringing mode, or checking your phone. I don't think we do that, but these are some of the things, we shouldn't do it. And looking around the room too much and avoiding the eye contact, because that doesn't show that you're confident enough. So, what you do is like, keep your hands in your lap or on your side. The best advice is to sit up straight and focus your attention on the interviewer. And if possible, before the interview, just ask someone to help you practice some of these answering questions and maintaining your professional behavior. And the next one we are going to talk about is about the virtual one. I mean, we all know about like how it's these days we have so many virtual interviews. So what are the do's and don'ts is you have almost the similar things here, but the the first thing is about you to test your equipment and video conferencing tools. Like, we have multiple tools like the Zoom, Microsoft Teams. So you should be familiar how to use them, how to on your cam, and how to mute yourself and this stuff. And next is about the body language. Just the way we spoke in the earlier slide, the body language says everything about you too. So don't slouch or lean back in the chair. Just sit straight. Don't play around with any paper on the desk or anything. So no matter where you are, it's all about, you keep straight, keep a good eye contact, and remain as calm as possible. Even if you are nervous inside, just show that you're confident enough and be calm. And what not to do is like, don't choose a distracting location. We know that, right? If someone is like, you have some background music or something, it is difficult for us and also the interviewer to concentrate on that set of stuff. So it's always better you avoid choosing the spaces with lots of things going behind you, like in a living area or like a coffee shop or anything. So it's, or you have to learn, if you couldn't find a suitable place, it's always better you blur the background and, you know, you have some quiet space so you can just have the complete attention with your interviewer and other things. And the next slide is about the networking. And this is one thing important in this graduate studies and all the stuff. What networking is generally a process of making some connections and some building relationships. So these connections can provide you with an advice and contacts, and which can also help you with an informed career decisions. So sometimes these networkings also help you with an unposted jobs and internship, because I read it somewhere. So that really helps in some ways. But how are you going to build these networks is like, you know, people generally join networking groups based on the single common point. I mean, for example, it may be a professional affiliation, or, you know, a college alumni group, or a club, or anything else. So for professionals like us, the best networking opportunities, where does they occur is during the seminars, conferences, and, you know, trade shows. Because these kind of things literally attract a large crowd of like-minded individuals. And during the poster presentations or like oral presentations, we tend to speak to the different people, and that's how we improve our networking and other things. So this networking, I think it also helps in finding you jobs. So it's always better how you network. And I think in the next slide, I can just talk to you the better ways about networking. Yeah. First and foremost, we all know that LinkedIn is the best way for networking. And the second thing is about joining associations or communities. And from the research background, when we are reading some particular papers or articles which we are interested or in the same path, you can always reach out to the corresponding authors, talk to them a bit, and many will be kind enough to reply you if you have any doubts or anything, and talk to your colleagues, and you attend the career fairs. I think there's one upcoming LVNL postdoc career fair, but I'm not sure whether the other labs can participate in this. And, you know, you can participate in some of the volunteer opportunities where you come across different groups of people, you can talk to them, and you can just increase your network. I'll just try to emphasize a little bit about LinkedIn. Personally, I landed in my current postdoc position through LinkedIn. I just applied in LinkedIn, and then the hiring manager from Sandia reached out to me, and that's how everything started. So LinkedIn has access to many organizations and associations. You can just follow them in and see their posts. This can lead like a more of a personal connection with the heads of the organization. And, you know, first thing is to create an account. We all know how to create an account. So you just create an account, but how to stand out from the crowd? So how does that? Add a photo, and you should add a location, and write a brief summary about yourself, because this is the first thing the recruiters will be looking at. And next thing is add the work experience, add education, and the skill set. Just list out all the skill set which you have gathered throughout your education thing, and this will really help you. And then you can start applying for the jobs. And sorry, the slide. Yeah. And the next one is, apart from LinkedIn, the other way for networking is to like join different associations and communities and become a member, like, you know, ACS, SIMB. These are the different conferences also, which we are, upcoming conferences which we are having. Like, you know, ACS is in August, and SIMB, this is here in California. It will be, I think this is in July 27th, 30th. When you start with attending this kind of conferences, you can just meet different people. And as I mentioned, during different presentations, you can talk to the people and always enhance your network circle. I think that's it about the networking. Yeah, thank you.
All right, thank you, Karuna. Thank you, everybody, for your attention. We have a quick survey if you're still online, and we would appreciate any feedback that you have for us for the workshop. And again, thank you to all our speakers, panelists, and moderator. And if you have any other questions, feel free to reach out to any of the presenters. If you registered, we have your email, and we'll send out video slides with all the links so that you will have access to all the items we discussed.
Thank you so much.
Thank you.